In English or in Portuguese? We were told to do it in English, but it's almost like a family here. But yeah, we can do it in English. So welcome, everyone. My name is Cília Rodrigues, and I'll be here together with Professor Mamed Carvalho, sharing this session. We have a good session ahead of us, I think. We will have some time for discussion in the end as well. Please take advantage of that and participate as much as possible. So as you know, this is the 5th conference, the redeSAÚDE, University of Lisbon, and it's really a pleasure to host this symposium on neurodegenerative diseases. As you know, what are the challenges of neurodegenerative diseases? We will be discussing these today with excellent speakers like Fede Herrero, who is here, right here, together with me, and also Professor Alexandre Mendonça, who is in Zoom and will be joining us a bit later as well. As you know, neurodegenerative diseases occur when nervous system cells, neurons, in the brain and in the spinal cord begin to die. And changes in these cells causes them to function abnormally and eventually result in cell death. And as this happens, an individual may first experience relatively mild symptoms, I would say, with problems with coordination or remembering of names and so on. But as huge numbers of neurons die, symptoms progressively worsen, and we will be certainly discussing this. In some cases, patients will lose the ability to walk independently, to think clearly, or generally function in the world. And ultimately, many of these diseases, as you know, are fatal. And what do we do? Well, in recent years, we have advanced and progressed a lot in understanding the underlying causes of neurodegenerative diseases. And as we uncover more details of these diseases, we are confident that we will come to a time where we will be able to treat and eventually prevent them. But as most of the major endeavors, the greater the resources we bring together, most likely the faster the progress towards our goal will be. And if we as a community, we want to avoid the dramatic impact of these diseases on our increasingly elderly population, we must all redouble our efforts now, for sure. And this is what we will try to discuss here today. And without further ado, let's move on, and I will give, there's another microphone over there, Professor Mamed Carvalho, to the first speaker with introduction as well. I will introduce Fred. Okay, so I'll do that. Fred, thank you for being here with us today. probably you may know FED already. His research focuses on molecular mechanisms underlying neurodegeneration and repair. He obtained his PhD in 2005. Then he did postdoctoral stays both at IMM and also in the Salk Institute, right? Yeah, in the United States. Then he eventually established his own laboratory at ITQB here in Lisbon as well, Oeiras. And since 2019, he moved closer to here, to the Faculty of Sciences. He's assistant professor there, and there he is developing cellular models of rare neurodegenerative disorders to study the principles and their principles specifically in living cells. So we look forward to hear you, Fed, today on molecular basis of neurodegenerative disorders. And thank you for being here with us.
Hola. Thank you very much, Cecilia. We are here in we are a small family here, so we all know each other. So thank you very much for the invitation and for the opportunity. Also, many of you already are probably familiar with my work and my line of work, but I will try to make an interesting talk today. Okay. So I was invited to talk a little bit about the molecular basis of neurodegenerative disorders, because neurodegenerative disorders are an emerging disease especially in the developing world, as the lifespan of people increases, because they are age-related. The World Health Organization is thinking that it's probably going to take over cancer as the second most common cause of death by 2040. So it's an important issue. Okay. Now my lab usually is integrated in this wheel, where we basically extract information from clinical sources, genetic causes, genetic mutations, histopathological features, clinical symptoms, and we use them to produce a series of here? No. Okay. So a series of models. We usually work with cell models, but we have collaborators, and I think the Lisbon area of neuroscience is very eager to collaborate, and they have a lot of model organisms for neurodegenerative disorders. So it's a very cohesive community. But we usually work with cells and with bioimaging methods. I will show you some examples of that. And then we try to use these systems to both explore new strategies, new approaches, new therapeutics, but at the same time using them as an excuse to understand a little bit more of the biology of the proteins involved, the genes and the proteins involved. Okay. I work at BioEasy and the Faculty of Sciences is my research unit, and basically there are several hallmarks. So there's two major paths of research that are currently being explored. One of them is personalized medicine, where you consider an individual as a particular case, and then you try to explore how this individual is and adapt the treatment to it. But there are other paths that is more traditional in science, which is trying to find common mechanisms to all neurodegenerative disorders that could be applied to all of them, or to some of them at least. Okay. So there's a lot of work done on the common hallmarks of neurodegenerative disorders. None of them has any cure, and the palliative treatments are very bad, usually with the possible exception of Parkinson. But even the Parkinson treatments are just palliative. They are just for the symptoms, and those do not do not stop the development of the disorder. They are all age dependent. They don't appear at any age. They have a pattern where age is an important factor. And there are other molecular and cellular factors involved, like oxidative stress, usually related to mitochondrial dysfunction, and the increase in transition metals that accelerate oxidation of biomolecules, neurotransmitter toxicity, lack of neurotoxic support, glycation, which is the addition of glucose or other sugars to proteins, non-enzymatically and specifically. And this is related to diabetes. If you have diabetes, you have more chances to develop Parkinson or Alzheimer or other neurodegenerative disorders. Inflammation is very relevant. A little bit of inflammation is protective, but when it's sustained in time, could be deleterious for the brain. And then the aspect that I'm going to talk to you most about, which is the one I've been developing my work in the last few years, which is the proteostasis mechanisms. So apparently in most neurodegenerative disorders, there are issues with the maintenance of protein balance, protein homeostasis. So either the proteins have difficulties to be degraded, or the proteins accumulate and aggregate in toxic aggregates in cells and tissues. No? So this makes neurodegenerative disorders, or most neurodegenerative disorders, belong to a big superfamily of disorders that do not affect only the central nervous system, but also the heart, peripheral tissues, etc., which are called protein misfolding disorders. Because in these disorders, what happens is that the proteins, the normal protein, the proteins mutated, they don't get to their final conformation, and they become sticky. They become more prone to self-associate, to interact with other proteins of their kind. And then they produce aggregates in the cells and tissues, and these aggregates correlate with the development of the disease and the degradation of the tissues, following eventually clinical symptoms, okay? So this is how it looks in cells. So in blue you have the nucleus, then you have in red the cytoplasm, and in green, yellow are the aggregates of proteins, in this case huntingtin, that accumulate in cells and tissues, okay? Now, the golden egg for industry and for most academic research in neurodegeneration is Alzheimer's disease, because it's the most common neurodegenerative disorder, and it accounts for 30 million people worldwide, in principle. Now, this is a little bit deceiving, because actually Alzheimer's disease it looks like a big disease, a big group of disorders with the same symptoms, okay, and the same histopathological features. Actually, 90 to 95% of the cases, we don't have a cause, and we don't have a clear, defined genetic cause for them, okay? They are sporadic. And these 10 to 5% of cases that are identified with a particular gene is not only one gene involved, there are several genes. So we could consider Alzheimer's disease as a disorder, as a group of disorders rather than a disorder. It's like cancer in this sense, okay? All right, the same happens with Parkinson's disease. It's very common, it affects elderly people, exactly the same. It's also very attractive for both industry and researchers because it affects a lot of people. But at the same time, most of the cases we don't know exactly why they happen, okay? And the cases we know, again, is 10 to 20 genes. They are different and they all share the symptoms and the histopathology, but to be honest, they are different disorders because they affect different genes, okay? So we have done some work on this, on these two disorders. Of course, it's difficult to avoid them as a researcher. Actually, we published recently a Nature Communication paper coordinated by Claudio Gomez, Professor Claudio Gomez at the Faculty of Sciences, about a chaperone, and we work on that. But my lab is mostly interested, it's a very small lab, and we try to focus on things, to try to find a niche that could be beneficial for society, but at the same time doesn't have so much competitiveness as in the big disorders, no? So we are trying to focus on rare neurodegenerative disorders. And what are rare neurodegenerative disorders? Any disorder that is not Parkinson and Alzheimer, okay? But of course, these disorders play with disadvantage. The fact is that they are all genetic, or most genetic, they are caused by one gene, and they are not in the same umbrella as a plethora of possible defects in genes, okay? Rare is a disorder that affects less than 1 out of 2,000 people. It's still not so infrequent. There are 7,000 rare disorders, not all of them are neurodegenerative, and they affect 30 million people as a group in Europe, similar in the USA. They don't have usually approved treatments, they are usually palliative, and most importantly, they all have a genetic basis which allow us to produce models. It's easier for us to produce a cellular model if we know the genetic basis of the disease, and then we can play around with that mutation and that particular protein, okay? This is Huntington disease. This is a disorder very similar to Parkinson, and although it is rare, it attracted a lot of attention because it has features very similar to Alzheimer's and Parkinson that can be studied from a cellular and molecular point of view. And I've been working a little bit on this disorder for quite some time. This disorder is caused by by mutations in a protein known as Huntingtin, which is a very big protein with unknown function, but only a small fraction of it is enough to produce the disorder in mice. And this fraction contains this polyglutamine tract, this polyQ tract there, which is a region where a CAG trinucleotide is repeated less than 35 times. When it's repeated more than 35 times, and this trinucleotide repeat encodes for glutamine, that's why the polyglutamine tract, when this is repeated more than 35 times, the people develop the disorder, yes, 99% of the cases, okay? So it's much more predictable than Alzheimer's or Parkinson, which you never know exactly if the people will develop or not, or when it's going to develop the disorder, all right? Okay, so this protein, Huntingtin, when you express it in cells, the normal protein has an homogeneous cytoplasmic distribution in the left, and it doesn't produce so many aggregates unless you overexpress it in big quantities, okay? But if you produce this mutation, this expansion of the polyglutamine tract, they produce large aggregates in the cytoplasm, all right? So these aggregates that look like balls in the microscopy pictures, if you pass through some super resolution-like application that was developed by a Portuguese researcher now in the Gulbenkian Institute, Ricardo Enriquez, you see that these aggregates are actually fibrillary. They are not balls, but they are kind of fibers that accumulate in cells, like crystals, and are really deleterious to cells. And I'm going to show you here a video. You check in the area where the circle is, of the dynamics of these aggregates, a little bit accelerated, okay? But you will see two aggregates fuse, and then you will see two big aggregates being formed in this cell, okay? And this sort of work we do a lot in the lab. So you see there the aggregates fusing, and then you are going to see appearance of the large aggregates in the cell that absorb any other protein, and then it gets with low dynamics, it gets very stable in the cell and very fibrillary, okay? But they don't really need to form aggregates to kill the cells, to be honest. The small oligomeric species also kill the cells. You see here there were not large aggregates, but the cells died with an apoptotic morphology. All right, so I'm going to give you some examples of what we are doing in the lab. No, we are working with, we are developing several models for rare neurodegenerative disorders. In this case, it's a disorder known as Alexander disease, caused by mutations in a protein that is part of the cytoskeleton in astrocytes, in astroglia. When this protein is mutated, it forms aggregates in the cell, okay, as you can see in the upper pictures. Now, we developed some tools to visualize single proteins in cells. We actually, each of these dots in the video below are proteins that are moving in the cell. And actually, we can see, by doing videos, time-lapse videos with this, we can actually follow up single proteins along a filament, for example, or in the cell. And these are tools that we are currently developing in the lab. Another disorder we are interested in, by chance almost, is spastic ataxia 8, another rare disorder, which is caused by mutations in the transcriptional repressor NKX62, which is involved in the development of the central nervous system. This protein is usually nuclear, is RNA binding protein, but when it is mutated, it loses in the left, the 141 mutant, it loses the nuclear localization and it spreads through the cytoplasm, or it can actually form aggregates in other types of mutations that produce the disorder, all right? Again, it's a rare disorder, but it's very interesting because NKX62 is very little known. It's not a very well-known protein. It's a little bit obscure, and usually it's compensated by other proteins of the family, but not in humans, okay? So it's a very interesting protein. And finally, I'm going to talk to you a little bit, very briefly, about this other model that we are developing by means of CRISPR. because we knew that succinct knockout, the succinct is a particular chaperone, is a protein, knockout produce a loss of function that produce RSACs, is autosomal recessive spastic ataxia of Charlevoix-Saguenay. It's typical of Canada, but is spread now through the world with low prevalency, but very interesting from the biological point of view too. Because when you knock out succinct, the intermediate filament network, the cytoskeleton, also collapses, and collapses in the juxtanuclear area, in those green spots you see there, in the bottom images. Those green spots are the accumulation of the cytoskeleton, which, if you see in the upper images, is spread through the cytoplasm. Okay, in blue the nuclei, in red is the mitochondria, and in green is the cytoskeleton proteins. So when you knock out by CRISPR in these cells this protein, the intermediate filaments collapse and get in the juxtanuclear area. So these cells are more sensitive to oxidative stress challenges. They are more sensitive to nutritional starvation, and they have some interesting properties at a molecular level that we are investigating at this point. And I think this is it. I hope you find it interesting. And only thank you for the attention, and the funding officers, the funding sources, and of course the people involved in this work, which were Fernanda Murtiñeira for the RSACs models, Pedro Peralta for the SPAC-8 model, Andrea and Joanna for the Huntington work. And thank you and the bioimaging facility of BioEasy, without whom we wouldn't be able to make those videos and those pictures. So thank you very much.
Thank you, Fed, very much. So now we have a few minutes, I think, for discussion. Any questions that you may have? Perhaps I can briefly start. So, Fed, this is very nice. Of course, there's a huge challenge in the approach that you are following, and that is there's so many cell types in the brain, right? And even within a given cell type, there's so many different populations. So how can you tackle that with your models on one hand? So how many are they? How do they convert to each other, you know, and to different populations? And also the correlates with the human disease. So how do you tackle these challenges within your models?
It's a difficult task because, well, I actually try to avoid it usually because it's too difficult. I usually work with cell lines, not so much with primary cell cultures. My focus is mostly on astrocytes and probably a little bit of microglia. Neurons are too much, there's a lot of work on them already, and I would like to know how the microglia and the astrocytes and the supporting cells are actually acting on it. But of course, they are also heterogeneous. They are not so homogeneous. So for now, we are trying just to create the tools and the models, and then the idea is to push them or to try to apply them into more heterogeneous settings, like primary astroglia, or even animals or brain slices. But cell cultures are a good platform in general because you can do screenings, the cells are immortal, you can do things. And if you have a good phenotype, for example, in the case of RSACs, if you have this aggregation that is very obvious, no, you can actually have a good starting point to check for possible modifiers of the histopathological features of the disease.
For sure. And the single cell technologies may also help, of course. Okay, any other questions or comments? Yep, please.
Thank you, President. My question is, because that dynamics of the huntingtin aggregates catch my attention in terms of the fusing and fusing events. My question is, do you think that these aggregates or crystals are more like an amylase state, or they have more properties of
liquid condensates?
Formed by liquid state separation.
Yes. So we are actually studying this now. We are preparing a paper on that with Huntington. So the big ones that you saw, the big ones that the last stages are the big ones. Those are actually crystals. They are amyloid. They are fibers, for sure. The other ones, there's a mixture. They are very dynamic still. So if they are fibers, they are dynamic fibers. But in general, depending on the conditions, in our case, we can modify that with phosphorylation and other modifications of the protein. They behave like liquid-liquid phase separation condensates. They behave totally like that. They are dynamic, they recover fluorescence when you do FRAP, they move around, they are flexible. You can, I didn't show you, but we have images of cells where the aggregate virtually expands and contracts, moving through the cell. So it's clearly a liquid-liquid phase separation system. We don't know exactly which are the limits, because sometimes we do see their fibrils, okay? So we don't know exactly how liquid they are, but they look like that, they behave like that.
Any other questions?
Thank you very much.
Thank you very much. I hope it's working. Is it working? I'm not sure. Yeah, it's same. Okay. So it's my great pleasure to introduce Professor Cecilia Rodrigues. She is very well known, so I don't have much job doing this. But anyhow, I would like to stress 2 or 3 things. One is a full professor of the Faculty of Pharmacy, and he's a very well-known, high-standard investigator with a number of very high achievements. And she has worked in the United States, in Cincinnati, and Minnesota. At that time, we had a link because she was investigating, I think it was your boss, the oxycyclic acid, and has an anti-apoptotic agent. And my patients at that time were very known. They were aware of these investigations for ALS. And some of them, because it was available in market, started to take the drug because it was a hope. And so we have that link, historical link. So now she's vice director for science investigation in this university. So great job again. So thank you very much.
Thank you very much. Can I have the first slide? This is not the first one. Okay, and let me see if I can. Okay, good. And now back again. Yeah, perfect. Okay. So, yeah, this is a challenge today, and I will talk a bit about the importance of collaboration and networking and transdisciplinarity on accelerating the drug discovery. So this will be the topic. I work on this at IMED, Faculty of Pharmacy, University of Lisbon, or I used to work on this. So can we move on? I think I have some problems here. Okay. Okay. Yeah, it's the previous one. So, the previous one. What do? We investigate pathophysiology and novel mechanism-based molecular targets for both therapeutics, novel therapeutics, and also biomarker discovery in many different types of diseases, including metabolic, degenerative, and neurodegenerative. I will talk about it today, and oncogenic diseases. And we specifically focus on mechanisms involved in cell signaling, metabolism, inter-organ communication, and pharmacology of bioactive molecules. So where should I point this? Because it's not moving. Okay. How do we do this? And we do this by integrating and by collaborating. So first of all, we use experimental and modeling technologies, including cell, tissue profiling, but also genetic and drug screening, with multiple preclinical models, including genetic, dietary, pharmacological, but also with patient data sets, samples, and models. We collaborate both within the academia, but also with hospitals and with industry to facilitate translation from bench to bedside. Okay, and here we are. This is our, I would say, circular research paradigm from bed to bench and back to bed. Indeed, as Professor Mehmet Carvalho was mentioning, we did start by investigating drugs that were in clinical use. These drugs were being used in hepatology. Then we evolved to research and development inhibitors of these molecules in neurodegenerative diseases. And here are some references to the impact, including more than 300 papers, global coverage patents on the topic, spin-offs that were actually from these studies, and including a phase 1-2 clinical trial. So on this, I would like just to leave here some highlights of the fundamental and preclinical studies that we did to really stress the importance of fundamental research and basic research. This is where everything starts. You cannot apply anything that you have not investigated in basic science. These are some examples. They have been published, and I will just move through them. And I would like to highlight some of the preclinical studies that we did. As I said, we have used the bile acid derivative. This was an approved drug. We investigated the mechanism of action, including safety and efficacy studies, both in vivo and in vitro. And with that, we published and were granted 5 global patents and 2 spin-offs. One of these spin-offs, Metacelex, is still active, quite active, in developing therapies to treat neurodegenerative diseases associated with cell death or cell function in general, including Alzheimer's disease. Then we went on to clinical trials, as I said. We had our own phase 1-2 clinical trial in amyotrophic lateral sclerosis. This was funded by Axcan Pharma in Canada. And these studies then evolved to other studies by other independent academic and industry researchers, leading to phase 2 and phase 3 ongoing clinical trials in amyotrophic lateral sclerosis, but also Alzheimer's, Huntington's, Parkinson's, and other diseases. And I just would like to leave here some of the data of a phase 2 trial in amyotrophic lateral sclerosis. This was published quite recently in September of last year. This is published in a very well-known journal, the New England. This is a group from Harvard. And what they show is that based on the use of this drug that we have discovered, on its potential to reduce neurodegeneration, they went on to this phase 2 clinical trial. This was a multi-center, randomized, double-blind clinical trial in more than 100 patients using the drug, and they clearly showed a slowed functional decline in these patients that correlated with survival and quality of life. So this was quite astonishing for us last year, at the end of the year. So then I would like to briefly mention, can I have the previous slide? Sorry, that was my fault. Okay, now I would like to mention that we went on to discover other new molecules. And we started with a very, I would say, big campaign of high throughput drug screening using semi-robotized systems that we have at IMED. Compound library screening, and I'm showing here some of the data. This is, of course, very general data, but I would like to highlight specifically one of the studies that is a study that is still ongoing, a very good example of open innovation between academia and industry. This is specifically with AstraZeneca UK, where we tested 250,000 molecules, a library from AstraZeneca for inhibition of a cell death mode, regulated cell death mode. We found 350 strong inhibitors. Some of them have known mechanisms of action, but many are novel and are now currently being further investigated both by AstraZeneca, Imperial College, and also here, University of Lisbon. We have done done computational studies as well and went on to proof of concept studies in mice, and I'm just briefly mentioning here some of the data. And this is key for even greater collaborations and transdisciplinary collaborations with other partners. And I'm just highlighting here one big consortium that we are involved in. This is liver investigation, but nevertheless, this shows the importance of being transdisciplinary on what you investigated. And this is a huge public-private partnership aiming to speed the development of better medicines for patients, a huge budget, a huge number of partners throughout Europe, including academia and including big pharma. And this is almost close to 100 partners. So really huge consortium, and it's only with this and this way that you can go and move further to develop new medicines. And these studies then led us to basic fundamental research, and I'm just showing here a brief example. This is a research on mitochondrial function, on the identification of a specific protein that controls mitochondrial fusion. This was in collaboration with António Zorzano in Barcelona, very well published in Cell, showing that this specific protein transfers phosphatidylserine from the endoplasmatic reticulum to mitochondria, and ablation of this protein really causes non-alcoholic steatohepatitis and liver cancer in vivo, with strong correlations in patients as well. Then another target that we have investigated recently on lipid metabolism, we identified the target we have shown correlates with metabolic liver disease in patients. This target favors inflammation, fibrosis, tumorigenesis, and controls metabolism. This is a target that may be important in many other diseases and not only in liver metabolism. And finally, can you target this? And the answer is yes. One of the drugs that we have used and discovered was employed in a model of liver disease to show that indeed you can reduce inflammation and fibrosis in obese mice, and that you can do this by controlling mitochondrial activity. And this is a key target in controlling mitochondrial activity, with correlates in patients as well. Okay, so what about going back to neurodegenerative diseases, and going back and forth between disciplines has really been enriching for us. And here I'm showing data on pharmacological inhibition of the same target that I've shown before in liver diseases, but now in Parkinson's disease. We have found a specific molecule, OXA12, that targets this protein that reduces inflammation and injury in Parkinson's disease mice, and has strong correlates in patients. Correlates with inflammatory targets, both in brain tissue, and I'm showing here some of the results, but also in patients in serum. And this is a collaboration with Professor Joaquim Ferreira at IMM and Hospital de Santa Maria to show the importance, again, of correlating what you find in vitro and in vivo models together with patients. Okay, challenges and unknowns. How can we better stratify patients? This is still a key question of any given disease that you may think of. Are there specific biomarkers? And what are the novel links and potential targets? And what I would like to perhaps stress here and highlight here, is the importance of multidisciplinarity, interdisciplinarity, and transdisciplinary activities. Because only this way we will be additive, interactive, and holistic in the approaches that we follow to solve world or complex problems like we have with these diseases. So I would like to finish here. Of course, thanking the people that have been involved in these studies, and all the collaborators, funding agencies, and also thanking you for your attention. Thank you very much.
Thank you very much for this wonderful communication, which is open for discussion. So I would like to ask you I have one question myself. the example you gave about the trial that was published in New England Journal of Medicine recently, and then a postdoc analysis was published in another paper in Muscle and Nerve. And really the phase 3 trial is starting, and I hope to be included and taking care of it. Is companies named Anilix, is an American company. But the question is, they combine two drugs, the sodium phenylbutyrate and your drug. And so when you screen, that is a little bit a question from someone ignorant about what is happening regarding this research. But when you are screening all these drugs, a library of drugs, in order to select the ones that can be very promising, my question is, do you have any tool to try to combine two different compounds in order to achieve a synergistic effect that could be even more effective in your model?
Yes, yes. So all these high throughput screenings that we do, we have the tools to combine molecules. And I really think that these complex diseases, I did not touch upon that, but I do think that these complex diseases with many different pathophysiological effects in place, you will need combinations, yeah. And I think this is a good point. So in that case, that study, they did combine something that is an antioxidant or somehow with antioxidant properties together with our drug. I say our, but it's not ours, unfortunately. But with the drug that we have discovered with strong potential to regulate cell function in terms of controlling cell death and inhibiting mitochondrial dysfunction. So I do think that's the way to go, and the tools that we have in place, we can combine drugs. That's true and very important, yes.
Okay, please.
I have two questions. The first question is technical. What readout did you use for necroptosis?
Okay, yeah, that's a very important question, because this is exactly the collaboration that we had with AstraZeneca. They said, yeah, you have to develop an assay for us. You have to develop something that we need. So they had the need. They came to us, and because in academia you can do that, you can try different methods and discover methods, we propose the method. And the readout is cell death. So we look at cell death. This is a phenotypic screen. We have a cell system, and we incubate the cell system with the drugs. In fact, they send us the plates with 384 plates, so these are big plates, yeah, with a large number of compounds. And there you can combine different compounds as you wish. So this is also the answer to the first question. And then the readout is cell death. So we develop an assay to specifically measure cell death. We actually combined a commercial assay together with something that we developed in the lab. The readout is cell death and necroptosis in this case. But yeah, we have other, we can develop other assays.
And the second question is, there's some issue that I discussed several times with my previous postdoc supervisor, which is the fact that for some time the pharmaceutical companies were a little bit obsessed, probably because of regulatory issues, on finding drugs that were specific to a particular target and everything. And my previous supervisor was exactly thinking about the opposite. Molecules that did a lot of stuff that was related to neurodegeneration and everything. How is the pharmaceutical industry now?
Yeah.
This is because the
Yes.
acid is that type of molecule.
Yeah, ursodeoxycholic is that type of molecule, but I think we should be open also there. And that's why we have decided to go to phenotypic screening. So we are not looking at a specific target. We are looking at a phenotype. And the phenotype may come from many different targets and probably a combination of targets as well. But then after you identify a molecule that is active in your screening, phenotypic screening, you can go on and deep in and identify the targets, which probably some of them are even not known. As I mentioned in our studies, a few molecules were targeted to some molecules that are known, but many, many more were unknown targets. And I think that's the beauty of it. And then you can discover something that is new and innovative. And I think this is the right way to move on. So it's important, the phenotypic response, and then you can deep in after you find something that is interesting, you can go on and identify the target. Because there's tools, structural tools and computational tools and artificial intelligence. We are going in that direction now. And yeah, there's many different tools that you can use, not only experimental, but also modeling with machine learning. Okay.
Thank you very much.
Okay.
I think we have to move to the third speaker, Professor Alexandre de Mendonça. I hope he's online. by Zoom. Professor Alexandre Mendonça is my colleague. He's a neurologist. We were residents about the same time, and then he started doing basic research in the Gobek Institute with Professor Alexandre Ribeiro, and then he finished his residency as a neurologist, and then he moved to a more senior position. He's a coordinator, investigator in the Faculty of Medicine, full professor, and now he's doing research in a more clinical perspective, and he's a top researcher in particular in Alzheimer's disease and memory impairment. So I hope he's online, and we are going to listen to his communication. So I think we have a small technical problem, I believe.
Não sei se me estão a ouvir. Não sei se me conseguem ouvir.
Yeah, the sound is good. Yeah.
Bom, então eu queria começar por agradecer aos organizadores e, enfim, cumprimentar os presidentes, agradecer muito as palavras do professor Mehmet Carvalho, enfim, que são com certeza também muito ditadas pela amizade que nos une já há muitos anos, e cumprimentar também os outros palestrantes, a professora Cecília e o professor Federico Herrera. Eu não sei se estão a ver o meu ecrã.
Nós estamos a ouvir bem, mas de facto não temos imagem. Estamos a ver o slide. Está bem. É o slide.
Okay. Well, so let's move to English then. And não sei se estão a ver de facto o diapositivo. Bom,
sim, o diapositivo está correto.
Ok, muito bem. So, the topic of today is Alzheimer's disease towards 2030. And if you can move. So of course, as you know, the problem of the memory complaints and Alzheimer's disease is becoming a very important problem nowadays. And in this horizon 2030, it is estimated that 78 million people would be affected with Alzheimer's disease worldwide, of course not accounting for many other people who have milder forms of cognitive impairment, even they are not demented. So I think it's a huge public health problem, and it will increase, of course, in the next few years. I think that when we are envisaging the future, it is important to understand also the past and to look at the past as well. And I will go back to 1988, so more than 30 years ago, and Carlos Garcia was the scientist, neurologist who started all the field of Alzheimer's disease in Portugal. And at that time he said that there were two main aims for the researchers in this field. The first one would be the discovery of biological markers that would, of course, allow a more precise diagnosis and an earlier diagnosis. So this would be the first aim. The second aim would be discovery of an effective treatment. And now, so more than 30 years afterwards, we'd say that the first aim was fully accomplished. So we can now diagnose Alzheimer's disease precisely and at a very early stage, even though we have no effective treatments yet to treat this disorder. So regarding the biomarkers, as you know, we have tests that can evaluate memory and other functions that are typically affected in Alzheimer's disease. And this is because patients have the marked atrophy of the hippocampus and the related internal temporal structures. These structures that have atrophy in these patients are connected to important regions in the temporal and parietal lobes. So you can see in the top right figure, I'm not sure I can point it out. In the top right figure, you can see with the PET-FDG that we might evaluate APOL metabolism in these brain areas. And of course, we have the amyloid, and the amyloid can be shown either in cerebrospinal fluid alterations in A-beta 142 peptide, or in the amyloid PET scan, which you can see on the left and low panels. And also, of course, you can look at the tau abnormalities, which are shown either in cerebrospinal fluid tau and total tau and phospho-tau levels, as well as in PET scan using selective radiopharmaceuticals to identify tau aggregates in the brain. So these are the biomarkers. And I would say that, in fact, there was a huge progress in terms of being able to identify the disorder. Of course, we know that there is a kind of a temporal sequence of these biomarkers. Probably the amyloid abnormalities are the first one to be shown in this path of Alzheimer's disease. And one of the most interesting discoveries in the last few years was that, in fact, the amyloid positivity precedes by about 20 years the symptoms of Alzheimer's disease. So this really opens an extraordinary window of opportunity for us to, could say, prevent the disorder. So we have about 20 years since the early deposition of amyloid and the symptoms. So really a great challenge and opportunity for prevention. Another consequence is that a substantial proportion of healthy people, and by healthy people we mean people that have no special memory complaints, and they do their normal lives, and if they undergo detailed neuropsychological testing, the performance is within the normal limits for the age and education, even though we know that at 50 years, about 10% of normal people defined as such will have already amyloid changes, will be amyloid positive, and that 65, 1 in 5 persons already have amyloid deposition in the brain or changes in cerebrospinal fluid. So this dramatically change, I would say, the way we can envisage the progression, the natural history of the disease. Of course, when you have big advances as being able to diagnose Alzheimer's disease very early, we also have then further questions. And one of the questions we're trying to tackle in our group was to see, well, it is represented by this saying from a patient who was diagnosed with early Alzheimer's disease. Yes, I hope for the best. It will definitely evolve, but I don't know. It will stay like that. Where will I be in the next 5 years? So the question of the prognosis of the individual patients diagnosed at early phases of Alzheimer's disease is certainly an important issue that has not been fully solved. We're trying to approach this question by taking advantage of the cognitive complaints cohort, which is a cohort of non-demented patients with cognitive complaints. That nowadays about 2,000 patients that have been followed by in the last few years, and so we're trying to see whether some tests that are part of the detailed neuropsychological evaluation can in fact predict how fast or how slow the patients with at initial stages of Alzheimer's disease might in fact progress. And we have so far observed that non-verbal abstraction, so higher performance in non-verbal abstraction, could somehow be associated with a slower progression of the disease, which is certainly interesting. But of course, we need to do more studies, and we are now trying with some collaborations with Sarah Madeira and Elena Heidge and at the Faculty of Science to use data mining techniques so that at the individual level we could, let's say, predict with a reasonable confidence interval if the patient would, for instance, progress to Alzheimer's disease dementia within 2 years, or the opposite. It would be probably stable after 5 years. So I think that this is something that must be addressed, and we're trying to do is. Another question that also arose after these possibilities of early diagnosis of Alzheimer's disease is that some patients who look like Alzheimer's disease are amyloid negative, and now we have this work under revision, trying to approach the question: what are these patients that are amyloid negative, and what is the outcome of these patients? Also, an important question that we are now trying to solve. So the first aim that Carlos Garcia identified more than 30 years ago was accomplished. We can diagnose reliably and early Alzheimer's disease. Unfortunately, the second aim was not fulfilled, and I think that this would be the aim for the 2030 horizon now. In fact, I just enjoy seeing this type of planetarium that aggregates drugs under clinical trials, and this was produced by Bengt Wingwat and co-workers back to 2010, and all those drugs failed to improve Alzheimer's disease, to slow the progression of the disease. And now we have this new planetarium in 2020 by Jeffrey Cummings and co-authors, and some of those experimental drugs, have already failed. So I think that this would be really the challenge for the next decade, to find an effective drug for Alzheimer's disease. It's still lacking this type of drug. And of course, just to mention that we have participated in a substantial number of clinical trials in the last few years, and of course, these also failed, as you know. Today, I will not tackle the question why these experimental drugs failed. I think that within the redeSAÚDE, next Friday, there will be, I will have a talk about these reasons, these possible reasons why the experimental drugs failed in Alzheimer's disease. But now I'd like just to briefly go back to some of the ideas that Carlos Garcia pointed out a long time ago, and because I think that these ideas are still very important today, and it could open, it could open, in fact, perspectives for development of drugs until 2030. One of the issues that Carlos Garcia mentioned was a question that was not solved, and it's not solved yet, which are the similarities between Alzheimer's disease and normal senescence, normal aging. And I think that this is a fundamental issue that has not been solved yet. In this slide, you can see it's taken from a very nice paper from Youssef a few years ago. And you can see here familiar landscapes from Alzheimer's disease. So you can see on the top right panel and right and left panels, you can see amyloid identified by immunofluorescence staining. And on the lower two panels, you can see congophilic angiopathy with polarized light on the left and immunohistochemistry on the right. And in the very lower and the right panel, you can see familiar images of neurofibrillary tangles, in this case stained by the silver modified Gallia staining. So these are landscapes familiar to all of us, but it's interesting to say that only the left upper panel is taken from a slide from a human brain of a patient with Alzheimer's disease at autopsy. All the others are not from patients with Alzheimer's disease. So the other images of amyloid are taken from old dogs, old and forgetful, by the way, forgetful dogs. And the neurofibrillary tangles are from an old, also forgetful cat. So I think that this raises a very important issue, which is what is the limit between Alzheimer's disease and aging? What can you learn also from studies in other species? And by the way, I think that the species that have been usually used for animal models, namely the mice and rats, are one of the species that are more resistant to amyloid pathology. So, and they have nevertheless been used in these models. So I think that this is a very important question. It's not solved, and it will provide, I would say, very important hints regarding the pathogenesis of Alzheimer's disease, and also, of course, the development of new treatments. And of course, at this point, I must acknowledge that several groups in Portugal have devoted very important efforts and publications to the aging and brain aging, of course. Another issue, another hint that Carlos Garcia provided was this topic of the heterogeneity of Alzheimer's disease. So, is there a pre-senile Alzheimer's disease versus a senile Alzheimer's disease? Are they similar? Are they different? So, what is there one type of Alzheimer's disease, or are there different types of Alzheimer's disease? As in fact, Federico has somehow already pointed out previously this morning. And for this purpose, I bring here the recent results from our colleagues in the Netherlands, and using cerebrospinal fluid proteomics, they can show that there appear to be 3 different Alzheimer's diseases, and not only one. One linked to hyperplasticity, changes in synapses, and another one linked to innate immune activation, and the other one probably more related to something like dysfunction of the brain barrier. So So apparently, according to these results, there appear to be at least 3 different types of Alzheimer's disease. Now, what we're trying to do now is with Bruno Bernardes and Sara Madeira and Isabel Santana and other colleagues, we're trying to replicate these results using a slightly different strategy. We're comparing patients with MCI due to AD, which is actually our patients with AD at very initial clinical stages of the disease, and comparing them with patients with complaints but without evidence or of amyloid or neurodegeneration. And at this point, we found out 165 proteins in the cerebrospinal fluid that are different between these 2 groups. So they seem to be somehow selective, particular for Alzheimer's disease. And applying these statistical methods, we have reached 3 clusters as well, as our Dutch colleagues did. We're not trying to see whether these clusters are superimposable or different from the ones that were previously described. But I think that the point here is that if there are different types, different forms, different Alzheimer's diseases, let's say, then we should move for a more personalized medicine, as it is now very popular to say, and it might be necessary to identify, to segregate the patients within these groups, and then try to do clinical trials separately, addressing these different targets. So just finishing to acknowledge people in my group and also the other colleagues and institutions and funding institutions as well. Thank you.
We are very grateful for this wonderful communication. It is open for discussion, so I ask if someone would like to raise a question. If not, I have one myself, which is more related to the public health, maybe. I mean, Alzheimer's disease is very linked to the aging process, but on the other hand, risk factors are the same as the vascular disease. So I wonder if you could correct some vascular risk factors, it could improve the probability of having Alzheimer's disease in old people. That's a very provocative and public health question.
Yes, I mean, it's a very interesting question. Contrarily to what people usually think, and in well-controlled studies, epidemiological studies, there's been a decrease in the incidence and prevalence of Alzheimer's disease in the last few years. I mean, at least in 2 decades, let's say. And although we don't have a clear explanation for this decrease in incidence and prevalence, I think that probably there are two causes for this decrease. One is, of course, that nowadays people have a different lifestyle, and so they are probably more active, probably they read more, and in aged people. And so there were important changes, improvements in lifestyle. And the other one is that people are, of course, more attentive to control vascular risk factors like, hypertension and hyperlipidemia and so on. So I think that this is a very important issue. So although we do not find, in the last decades, we not find new drugs, but we could decrease the incidence and prevalence. And I think these are really good news in the sense that we might do something to prevent, at a certain point, to prevent, Alzheimer's disease and other forms of neurodegeneration. So I think this is really good news, and of course, it has been difficult to identify RCTs, randomized controlled trials, to identify without any doubt what measures are important to decrease the risk of Alzheimer's disease. But I think that these results are certainly encouraging in the sense that we should improve our lifestyle and improve our health care from this point of view, with both significant outcomes, both at the cardiovascular level and at the health brain level as well. Also, I'd like to now to approach a different, to mention a different related issue, which is now we know quite a lot of things about epidemiology of Alzheimer's disease, and now it's needed to refresh this knowledge regarding amyloid positivity. So I think that it will be very important to do longitudinal studies in the community and try to address what factors, what risk factors and protective factors are involved in the amyloid buildup in the brain, and try to modify them. I think it's another epidemiology, finer epidemiology that we are facing today. Of course, it will be difficult because the costs would be enormous, and but I think it would be a priority at this moment.
So thank you very much. If there is no other question, I think. Thank you, Alexandre, for your wonderful talk.
Muito obrigado.
Obrigado.
Bye.
So it's my job now to just in 1 minute to summarize what happened today, and so it was a wonderful moment, and we learned a lot from aggregates and how they move, and which is amazing, that movement, the dynamics of the aggregates, and then we jumped to how you can accelerate discovery of new drugs in the very large consortiums, but also by these libraries research, and how we need to know more about these drugs that can be effective, because the mechanisms of this effectiveness is not well known, how you can go and to find new compounds for treating these terrible disorders. And Alexandre raised two important questions in addition to many others more strictly related to Alzheimer's disease. One is about how you can manage this difference between generation and senescence, the aging process that we are involved, and how we have to be careful about this precision medicine or personalized medicine approach, because maybe the response to different treatments is very, in some way, variable between people, because the disease features are not similar, exactly the same in everyone, and so even suffering with the same condition. So these disparities, these details can be very important, and that's something that we need to think more and was approached here. So this is my summary. I'm very happy to be here, so thank you very much, but I'm going to.
Okay, I think we should close. Thank you very much for your presence. I thank also Professor Mehmet Carvalho for being here with us and moderating the session. Of course, Fred, Professor Alexandre Mendonça, away from us, but close to us, of course. And we should move on now to the other room because there's another session probably already starting. Thank you, and I hope you continue enjoying the day. Thanks. you.